Today we are going to introduce you about CRISPR-Cas9. So we know that we can use bacteria to produce insulin, and scientists are trying really hard to turn pigs into an organ donor candidates for humans. Also, we know that we can breed goats that can produce silk protein in their milk. And if you don't know this, make sure you are in the class. So how does it work? You know that we can't just inject it into the mature organism.
Okay, how to do that? First thing first, we need to make a whole cell have the sequence of DNA. That is, we need to inject the plasmid into the cells before it starts to divide. The cell often be a zygote or bacteria. And the most important thing is that before the injection, we need to determine where this gene of change should be presented. Okay, if you don't do that, you might have to suck the blood out of an innocent animal, or even kill it just for the organ you want. I think nobody would like to do that. So after we find the location of the sequence, we need to design a mechanism which can recognize this sequence and insert the plasmid with the gene we want to present it. That's right, the mechanism is CRISPR-Cas9.
CRISPR-Cas9 method composed of CRISPR RNA, transactivating CRISPR RNA, and Cas9 protein. Transactivating CRISPR RNA can generate hydrogen bound with CRISPR RNA and form guide RNA. Transactivating CRISPR RNA also have a function to combine with Cas9 protein. And then Cas9 protein will try to find the 10 site, which is a 3-code sequence on the DNA. After the combination of these two things, CRISPR RNA will try to match the sequence on the DNA. If the DNA sequence is complementary to the CRISPR RNA, then Cas9 protein will generate double-strand break on the DNA.
Cool.
Use these things, we can design a single-strand RNA. We call this single guide RNA, which has the function of transactivating CRISPR RNA and CRISPR RNA. We can just alter the CRISPR RNA function site sequence to determine where we want the sequence of DNA to be broken down.
Cut it again.
After the breakdown of the DNA, we can just give the cells with a plasmid, which contains the gene we want the cells to express, and let the cells repair with this given plasmid. We call this process homologous recombination. Here's our DNA. And of course, it may just connect with original breakdown place. It's called non-homologous end joining. According to a postgraduate in academia Seneca, 60% may successfully repair with the given template. Thank you.